Okay, so this is my great pleasure to welcome today in this Philosophy in Biology and Medicine seminar series. Rick Michaud. Rick Michaud is a fantastic biologist, an expert in ecology and evolution, and a professor at the University of Arizona. And Rick is an expert on evolutionary transitions, including transition to multicellularity. And he's been working on different volvocine models, on some volvox in particular, over the last years, decades. And Rick is very, very engaging and very smart. And I invited him in Paris at IHPST a few years ago, and I really loved the interactions we had, both his talk and the discussion. So I had thought for years that he would be a perfect speaker for this PhilInBioMed seminar series. And this year I sent him an email and he said, yes, I'm happy to do it. So we were very enthusiastic about it. So today is the day. Rick is going to talk about reorganization of fitness during evolutionary transitions in individuality. So, Rick, thank you so much for accepting our invitation, and the virtual floor is yours.
Good. Thank you, Tomas. And well, what a pleasure to be here and talking to you from Tucson, Arizona. It's a beautiful day here, beautiful morning. And I trust it's equally beautiful where you are. You know, so much beauty in the world, and so much complexity. And the challenge is to explain it, through evolution. And I'm not going to, of course, explain it all today. What I hope to explain today is the familiar fact that the living world is organized hierarchically, and it's this aspect of complexity that I want to address today. And I'll image the hierarchy of life in several ways. What I struggle to do is to present the hierarchy without giving a progressive view of evolution, that evolution always tends to complexity. I want to avoid that. But nevertheless, there have been these dramatic jumps in complexity. The living world began simple, so it's not surprising that it became complex. What is surprising is that it became complex in this hierarchically structured way. And I've imaged that here in terms of nested levels of individuals, and then this kind of sweeping arc of complexity, increasing complexity from DNA molecules to interacting groups of molecules, molecular replicators, to a simple bacteria-like cell, to a eukaryotic cell, to a multicellular organism, which is what we're going to be talking about mainly today, then a jump to eusocial organisms. Anyway, so that's the challenge today, is to explain this hierarchical organization. And I want to do it through two central concepts. One is fitness, and then the other is individuality. Individuality for me is not just a descriptor of the world, it's something that really emerges out of the dynamics of natural selection. And I want to explain how that may work. So anyway, the organization of my talk, just to make some general comments about order and fitness. I've already made a few of them. And then I want to talk about the transition from unicellular to multicellular individuals. I'll introduce you to this beautiful group of organisms, the volvocine green algae. And then I'll talk about mathematical models I've used to explain evolutionary transitions using multi-level selection. And then I'll talk about development, how multi-level selection of genetic modifiers of development shape development, so that the group can become, under certain conditions, becomes an individual. That's really the central question that I work on, is how do groups of individuals become new kinds of individuals? That's my central question. And then I'll also talk about life cycles at the cell level, the group level, how the group life cycle can emerge out of the cell life cycle. And then I'll talk about some recent work we've been doing about translating this research into the classroom, because we think that kind of the social constructs that we use in understanding ETIs, cooperation, conflict mediation, are familiar to students. You know, they're social beings, and so they have an intuitive understanding of the benefits of cooperation, but the temptation to cheat. And so this can be used, I think, in a more engaging way to teach biology. And then I'll conclude by just some general remarks about individuality. Good. So just a few comments about order and complexity in biology. A lot of our interest in biological order comes from these remarkable adaptations that organisms have. They even appear to be designed by some external agent. This was confusing for many years, but we now know that the self-organization of populations can create adaptation, and central to that is a concept of individual fitness. Another kind of order in biology is the species. You know, the world doesn't come to us just as a shmoo. There's distinct kind of groupings of organisms and gaps in phenotypic space. I'm not going to talk much about that. I just want to acknowledge that's a very important kind of order. The other kind of order that I'm really going to focus on is individuality. We need to count things in evolution, right? So we count individuals, but under some conditions we count cells. Under other conditions we count whole organisms. And so we know that evolution has created different kinds of individuals. And so these different kinds of individuals are represented in the hierarchical organization of life of these different increasingly complex individuals. Good. So the goal of our work is to reconcile fitness with these transitions in individuality, the concept of fitness. How does fitness emerge at a higher level when it's not present there, or there is not present there much initially? Just a few, since I know there's a philosophical audience there, I just want to talk a little bit about explanation. Not that I'm a philosopher, I'm not, but you know, there's recognized to be, you know, theological or teleological explanations. Things have properties and exist because purposeful agents make them. So we're very familiar with that through artificial selection and through, you know, the design of watches that we find on the beach and so on like that. Purposeful agents make them. Another kind of explanation is mechanistic. Things have properties because of their parts. And then Darwin really introduced, I think, a totally different kind of explanation involving population thinking, where populations self-organized via the dynamics of the population. And this self-organization can create adaptations and this that exist in the world. And the fitness is the central concept. Lewontin once remarked that fitness is the only uniquely biological concept, that everything else is chemistry and physics. And I think he had something there, that really it's this remarkable property that of adaptation and the resulting fitness that it needs to be explained. So evolution should explain fitness. Well, how do we do that? So in my book many years ago, I adopted this criterion for understanding fitness, that we need to explain the origin of it during the transition from non-living to living, that we should explain how fitness transitions from different levels, among different levels during the creation of new units of evolution or new evolutionary individuals. And finally, we should understand how it's used in evolutionary explanations. But today we're going to talk just about this problem of the transitions and units of fitness. How does that occur? Okay, and the other I've already remarked on the hierarchy of life. History shows that the units of evolution have changed over time, and that life has this nested hierarchical structure. And how did it arise? That's what we're trying to ask today. How do new levels or individuals arise from groups of existing individuals? Or to phrase it the way I like to phrase it, how do groups of individuals evolve into a new kind of individual? That's my main question today. Okay, just a little bit about terminology fitness. What do I mean by it? I'm just using it in a very simple sense. Organisms or individuals have to survive and then reproduce. So the simple representation of that is just survival times reproduction. Survival and reproduction always trade off. That is, you can't have maximum survival, maximum reproduction. Effort of one decreases the other. So I'll talk about trade-offs. I'll talk about genes that control these trade-offs or that use these trade-offs to either turn off reproduction, in stressful environments. You want to just get through the environment. So you want to turn off reproduction so that you survive, so that your survival increases. All living things have to have this property where they can regulate their components of fitness from simple molecules to complex colonies, I would claim, or I would hypothesize. I'll be very interested in germ cells that specialize in reproduction, or more accurately, somatic cells that turn off reproduction, because that's altruistic in a sense. But anyway, germ cells specialize in reproduction, somatic cells specialize in survival. Altruism is a form of cooperation that is costly to individuals but beneficial to the group. The costs of altruism decrease fitness of individuals while benefiting the group. And because of this property of altruism, I like to speak of altruism transferring fitness from the individual level to the group level. I've been criticized on this notion, and I understand the points being made. I don't think fitness is a conserved quantity, but I think there are altruistic traits that do exactly this transfer. They benefit the group, but they harm the individual. So I use the word transfer for that. I like to speak of fitness reorganization, which involves this transfer of fitness from lower levels to higher levels, but also the specialization of lower level units in the fitness of higher level units. Multi-level selection occurs in a group structured population in which there is both within group selection and between group selection. And then evolutionary conflict is when selection at the different levels go in different directions. For example, between group selection may favor cooperation, within group selection may favor defection. Finally, conflict, or another word, conflict mediation, are traits that restrict within group selection or enhance between group selection. And finally, I just use the word evolutionary individuals. They possess the Darwinian properties, the Lewontin conditions of heritable variation and fitness, along with traits that mediate conflict by decreasing selection at a lower level. And in that way, complex adaptations can evolve at the individual level. I'm really interested in that question of just the evolution of complexity, complex adaptations. And I would claim that only evolutionary individuals can do that. Okay, so multicellularity is a complex trait, right? And so following Darwin, we want to reduce this jump in complexity from unicellular to multicellular organisms to a set of possible steps, each advantageous in and of themselves. So we're going to do this in three ways. We're going to first use multi-level selection involving the steps involved in multi-level selection. Then we're going to use steps in development. And then we're going to use steps in cell cycle regulation, how to get from the group life to the group life cycle from the cell life cycle. And we're going to use the volvocine algae for many of this. And I'll introduce you to them in a second. But first, multi-level, and I guess the slide just says what I said. These are the bullets for what I just said. Good. Okay, so the volvocine green algae, they're a spectacular lineage of organisms. We find them in the desert Southwest, but they're found around the world, and you can find them in ponds like this in southern Arizona. Here we are, some students collecting them. They range from unicellular Chlamydomonas, just a simple single cell with two flagellum, And then there are more complex, simple groups of just four cells, Tetrabena, basiclamis. They just have four cells. They're claimed to be, this four-celled species is claimed to be the simplest multicellular organism by some. Then there's gonium. I'll talk a lot about gonium. It's an eight-celled colony. Um, Most of our current work is on gonium, but I'm not going to talk much about that. But it's just an eight-celled colony. I'll talk a little bit about it. And then there's this beautiful critter called volvox, in which you see inside, this is a mother that's pregnant with her daughters here that are going to hatch out soon. And then in addition, there are these tiny somatic cells on the exterior of the colony that provide motility while these daughters are being made on the inside of the colony. So this is just some images, again, just Chlamydomonas, single-celled, gonium, an simple eight-celled colony that's kind of flat, not spherical. Uterina, which is spherical, but has all the cells are identical. That is, there's no cell specialization, but it does have this interesting spherical form. Another species, Pleiadarina, which has smaller somatic-like cells, and then general purpose cells. It's interesting in this group that soma evolves before germ specialization. And then again, volvox, and we see this is a juvenile with just a germ cell on the inside and a somatic cell. These germ cells will divide to become these daughter colonies that hatch out. Good. Okay, so we have this beautiful complexity in these algae, and we want to reduce it to a small set of steps, each advantageous. So multi-level selection, we want to talk about the origin of the group, the evolution of cooperation within the cell group, the increase in size of the group, which sets the stage for conflict and how that conflict is mediated. And of course, if it's not mediated, the colony just goes back to it loses the cooperation. Then we talk about the specialization of different cells in the group and the increased integration of the cells in the group, and ultimately group reproduction. And at that point, when you have specialized cells, in a sense, the group is indivisible. That is, if it has specialized soma, specialized germ, those cells cannot live on their own outside of the group. They require the context of the group to live. And so the group becomes indivisible. You can't chop it up and have living parts. Okay, and so then there's developmental steps. I'll talk about this and but I'm not going to. But I'll delay that. But these are genetic developmental steps that have been hypothesized by a famous David Kirk as the developmental steps that create individuality. Tomas, I just got a message that my internet is unstable, but I'm just wondering, can you hear me? Are you following?
Yes, we are following, but it has become very slow in the last 30 seconds. So if you have two networks, maybe switching networks could be a good idea.
Yeah. Okay, let me just do that right now. And we'll hope that solves the problems. Good. Okay, so we're back up here. So multicellularity is a complex state. Okay. Is this any better? I'm just curious.
For now, it seems to be quite the same, at least where I am.
Okay. I'm sorry. So we'll try this. If it becomes untenable, then maybe we'll switch back. Maybe that was just a small, as the desert heats up in the day, I'm on a wireless network and it can create trouble. But anyway, how we doing, Tomas? Should I keep going with this network or back to the other one?
Um, Oh, okay. So Dan says that from Rhode Island, the second option is better than the first. And indeed, you seem to have become yellow on my screen, so it's better than the red. So we should continue with this one. Thank you, Rick.
Okay. Thank you for that information. Don't hesitate to interrupt me. I'm I'm not in an optimal physical location right now for internet. So okay. So we're going to first focus on multi-level selection. Great. And so we have these models that we've developed based on the algae, but are kind of just motivated by the algae, but we think they apply in general. Propagules are made that then divide through mitosis to produce some kind of adult. And we have parameters for this, how much time, what is, at every stage of cell division, there can be a mutation, and that mutation affects the level of cooperation that the cell may engage in. And then the group fitness is calculated as a simple result of how many cooperative cells there are in a group. It's not an aggregative function. There's ways we think the cells interact that inform how we calculate the group fitness. But I'll say more, maybe I'll say more about that or not. So, but again, central is cooperation. So a cooperative cell, it replicates more slowly as a result of putting energy into helping other cells in the group. But the group is more functional as a result. And defecting cells are just the opposite of that. Okay. And so we assume a simple Mendelian beanbag genetic architecture here. Two alleles cooperate and defect. And then and we embed this locus in the context of another locus, a modifier locus, that will affect aspects of development. And the modifier locus, capital M, may sequester some cells at some stage into a germline where the mutation rate's lower or something. It may create policing. It may determine the adult size and determining growth. There's all different kinds of modifiers that we've studied with this kind of two locus population genetic framework. And anyway, we go through a process of calculating the adult, and I won't go through that now, but to just recognize that we've studied different kinds of conflict mediation, programmed cell death, and so on, self-policing. We've done a lot of different papers on this using different kinds of conflict modifiers. Not just want to recognize that, don't want to go into any depth. Anyway, we write down standard two locus population genetic equations, and the interpretation of this is that cells or that organisms or that groups that have the D allele and the no modifier, they're just basically single cells. There's no cooperation, and there's really no organism. And then the thing we're interested here is the evolutionary equilibria where we have a group of cooperating cells, but no higher level function, no modifier. And then we're interested in the transition from this equilibrium to this equilibrium 4. Sorry. And and we interpret this equilibrium 4, which has cooperation, but it has a new modifier, which enhances cooperation. We interpret that as an individual because it has these Darwinian properties, but it also has within level change mediated. Okay, so we're interested in this transition. Mathematically, we can define all that and study it and make conclusions. And what we find is during this transition, we see that the heritability goes up and the within organism change goes down. Let me just pause. How are we doing on the mechanics here? Is the audio okay and the visuals okay?
For me, everything is okay. If there is any problem for anyone, you can use the chat either to everyone, not just to me, if you think there's something to fix. But for me, Rick, everything is going very well.
Okay, thank you. Good. So we've also studied this with using reproductive effort models of division of labor, and as reproductive effort becomes specialized, represented here is the variance in reproductive effort. The group fitness goes up, self fitness goes down. Anyway, so we like to recognize here that fitness is being transferred from the cell level as organisms, as the ETI continues to the group level. Okay, so we speak of the transfer of fitness. And so these are just general statements. So the modifiers have developed. What are we watching here? We're watching the evolution of these modifiers, which are neutral, but they hitchhike actually with the more fit chromosomes. So we have what we have in the genetic sense, if people are familiar with population genetics, we have a mutation selection balance because there's mutation constantly taking cells from being cooperative to being selfish. So that creates a mutation selection balance, and there's variation now in cells in the population, and the modifiers can hitchhike with the more fit C chromosomes, more fit at the group level. They can hitchhike with that. Their effect of making the group more, having more fitness variance at the group level. So we have this hitchhiking going on. But anyway, so the individual is a cell group with these higher level functions, such as germ soma differentiation that mediate conflict among cells. In effect, the modifiers take multi-level selection from what's called MLS1 to MLS2. So the wrap up here, cooperation benefits a group, it leads to conflict, conflict leads to conflict mediation, and the modifiers of developmental processes evolve to reduce this conflict or can evolve. And when they do, individuality emerges at the group level as the heritability of fitness at the group level increases. So we can measure all this, plot it all out for these different kinds of models. Good. Okay, so that's multicellularity is a complex trait. These are the steps here of multi-level selection. Let's look at the steps of development. So we're not going to spend a lot of detail here, but there are these 12 steps that David Kirk had hypothesized in his work. These are genetic developmental steps that have to occur. And you'll see some familiar things here like germ soma division of labor, but then you'll also see things like increased volume of ECM. This is this space filling material that helps create the size of the colony. You'll see things like genetic modulation of cell number. That's very important. Have to control the replication tendency of cells. And if you don't have ways of doing that, then it's hard for cooperation and integration to evolve at the group level. So we're going to focus on this now. And the way we do that is we do it through phylogenetic reconstruction. So we have here the phylogeny of this lineage of volvocine algae. And in color here are the different multicellular forms. And then these are, and the outliers here are the unicellular species. And I mean, one thing that's interesting about this group is that they evolved, this interesting complexity, two specialized cell types, this beautiful spherical organization where the somatic cell and this spatial structure where you have somatic cells on the outside and the germ cells and the daughters are on the inside. And that's as far as it went, or at least so far in the 200 million years that this has been going on. The road to higher plants is elsewhere. So we find this lineage interesting because there was this interesting spurt of the evolution of complexity, but it only went so far. And why? We'd like to know not only why the complexity evolved, but also why it appears to have stopped evolving. And we think it's because of of the way they evolved soma. I mean, I'll just do a sidebar here. And we're going to get to this at some point in the talk, but the soma that they've evolved is not like the soma in our bodies. It's not like the soma in our bodies has this unlimited replicative potential, right? The well, maybe with the exception of the brain, but in our stomach, you can always produce more cells, but in Volvox, that's not the case. That is, once the soma evolves, it's a dead end in development. It can't go on and replicate. So the somatic cells are kind of a starved lineage of cells within the organism. They can't go on and replicate. It's terminal differentiation. Anyway, that was a sidebar. Back to this phylogeny. So we see the multicellular forms here, the unicellular. And listed here are these developmental genetic steps. The black boxes are losses of them. White boxes are gains of them. You're not supposed to understand all this. I'm just going to make a couple bullet points about this mapping. So we map the steps onto the phylogeny as when they occurred and so on. So what we find is that sterile soma evolved 3 times, and that that was sterile soma, and then it's lost twice. Okay, so another reason we like this group, or we feel we can, informs our thinking about complexity, is that complexity can increase in the sense of somatic cells evolving, but it can also be lost. And so there's not a progressive march to higher complexity in this lineage. And likewise, reproductive cells, they've evolved twice in this lineage and lost once. The key innovation, if we go way back here to what created the multicellular group, is 2 traits evolved at this point at the beginning. One is the genetic control of cell number, and the other is a transformation of the cell wall into this extracellular matrix. And basically, we interpret the matrix as being a way that the cells cooperate, a way of them sharing materials and so on. And we interpret the genetic control of cell number as a way of mediating conflict. It's the most basic mediation device. You control the rights of certain cells in the body to divide, or the organism does, or there's this higher level control that gives rise to the individuality eventually of the group. Okay, so we like to interpret the evolution of individuality through this lens of cooperation, conflict, and conflict mediation. So in summary then of this part of the talk, evolution is not a progressive march to greater complexity. There's multiple gains and losses of key multicellular traits. And there's periods of stasis. You have these long periods, these long, 200 million years where the body form is basically staying the same. So you have periods of stasis here, and then you have rapid diversification, like here, in like just maybe 50 million years. You have a rapid diversification, then stasis. So we have that feature going on. The temporal mode is changing. And then we have these cycles of cooperation and conflict mediation. Good. So a little bit more about reproductive altruism, but I think I'm going to skip through this. This just kind of drills down on a genetic gene that we've been studying that makes cells somatic. Let me just get a gauge of where I am, because I want to wrap up in about 10 minutes here. How are we doing on the technology? Are people hearing me?
Technology is great and time is great, Rick, because we started almost at 10 past. So you really have until now 9 at least. So do not be too worried, I think,
Oh,
about the time.
yeah. Okay, well, okay, so I'll just dive into this a little bit. So in these algae, we have the cell cycle. Chlamydomonas is a single cell organism. It grows, then it and gets big, and then it divides, and the cell divides to produce daughter cells. And there's this trade-off between the microtubules that are needed for um, the flagella and the microtubules that are needed for the mitotic spindles. So when a cell's dividing, it can't have a flagella. So there's this important trade-off that you can't divide while you're motile. And so this life history trade-off constrains the life cycle of the single cells. And so when we fast forward that life cycle into Volvox, we have now cells that are specialized in the flagella stuff. They keep their flagella, and then we have germ cells that lose their flagella. And the presence of flagella in the somatic cells allows the germ cells to divide while the whole colony is motile. So the colony can break through the trade-offs that govern the life cycle of single cells. And if there's specialization, right? If there's cells that specialize in keeping their flagella, but they can't reproduce, and that allows germ cells to divide while the colony is motile. I'm just repeating myself. Okay. So there's a gene that controls this progression through the life cycle in Chlamydomonas. It's a master regulator of a chloroplast biogenesis. A lot's known about it. I won't go into that, but it keeps cells small when it's expressed. It keeps cells small by kind of starving them. Basically, not having good chloroplasts. The chloroplasts are not developed. They can't grow. This is it's a plant that lives in the sun, needs the sunlight to grow. And so not being able to grow, it can't divide. So it's just a starved cell. It keeps its flagella and it can help the colony move. So we speak of altruism here, that it's a gene that, benefits the group, but it restricts the reproductive component of fitness of the cell. And in Volvox, it's expressed developmentally. So in clammy, in the single cell organism, it's expressed environmentally. So when a unicellular clammy is in a stressful environment, it doesn't want to try to grow, because if it does, there's just not enough resources to allow that. So it blocks the progression through the life cycle until there are resources. So the cell can kind of hang out until better conditions return. So we call this a life history gene at the cell level, and it's co-opted to be the gene at the group level. So, just to be simple here. So you have these genes that they control survival and reproduction. And in stressful environments, why all organisms have them, because in stressful environments you have to turn off reproduction to get through the stress. So we expect all organisms to have them. Clammy has it, but the same gene then can be co-opted in a spatial set to turn off reproduction in the group, and that's where soma evolves from. But I've gotten way ahead of myself here, but maybe that'll help me get through this quickly. So now, when there's a mutation in the Reg gene, all the cells now try to reproduce. We end up with a colony that's just a mass. It's like a tumor. It's like a cancerous mass of cells. All cells are reproducing. Nobody is doing the flagella stuff. No, the colony's not motile. It sinks to the bottom. This is just how we. So anyway, so we're interested in life history genes and stress. I've already said this in my commentary in the last slide. Okay, and so this is just a diagram of this picture. This is just a cartoon. Okay, in a unicellular individual, we have these genes that are on in a stressful environment. And in that situation, the cell survives better, but doesn't try to reproduce. It's expressed in a temporal environmental context. So that's in a unicell. Those same genes then can be used in the group in a spatial context, that same gene or a homolog of it can be turned on, and in the somatic cell, it does the same thing it did back here. It turns off reproduction, makes the cell somatic. So that's our basic hypothesis for the evolution of soma in this group. But we think it's a general, so we often wonder why we're studying Volvox, and well, we think this part of it is general. Not all organisms have Red J or RLS1, but they're going to have life history genes that can be mobilized to tease out the components of fitness in the group and specialize cells. So we think this point is a general point about how reproductive division of labor can evolve. This is just a whole gene family that we've been studying of these Reg A cluster genes. I'm not going to say any more about that. We're very interested in the history of this gene family. And let me pause there and just say that the genome of Volvox and the genome of Chlamydomonas, so the genome of the most complex member of this lineage and the genome of the simplest member of this lineage are basically the same, except for three gene families that have diversified. One is this Reg gene family that creates cellular specialization. And the next gene family we're going to talk about when we talk about the group life cycle are the cyclins. But we'll get to that in a minute. Okay, so the wrap up on this is that cooperation increases the fitness of the group, decreases the fitness of cell. The group formation can translate these lower level fitness trade-offs into cooperation and genetic basis for reproductive altruism in the groups may arise from these life history genes that are stress response genes in the unicellular ancestor. We think that's a general point, not just this lineage. These stress response genes are good candidates for fitness reorganization in the group developmental context. And this gene family in the algae that we study originated in non-differentiated groups where it was involved in a stress response. Okay. So now the third way that we break down the steps to complexity in this group has to do with the life cycle. So I want to jump into that right now and talk about the origin of group reproduction. In their seminal work, Maynard Smith and Zathmari recognized that one of the criteria in evolutionary transitions is that entities that were capable of independent replication before the transition can replicate only part of a larger whole after it. So this is basically the problem of group reproduction. Reproduction at the group level is, really a central issue in the emergence of this new level of individuality. MLS theory, multilevel selection theory, distinguishes between these two different kinds of multilevel selection. And in MLS2, the lineages that exist are at the group level. And so, again, that recognizes the importance of group reproduction. So quite simply now ask the question, how can groups acquire the capacity to reproduce? That's the origin of a group life cycle. Well, we're going to use the algae to think about this. Okay. So this is the normal cell cycle in Chlamydomonas, the unicellular ancestor, well, thought to be similar to the unicellular ancestor of this lineage. What does a cell do? It grows, and then it divides, and then the daughter cells separate. So we recognize immediately in this lineage, there is a transient group stage, right? There is this transient group stage. And so the simplest way to create a group, the origin of the group, can simply be a result of the failure of separation of these cells. Okay, so and this has been recognized. I think Bonner, in his little book, posited this as the most frequent or most common way to get a group. Okay, so if we have the failure to separate, so we have now these four cells, well, what can they do? Well, if they were in their past program, they would just start growing. So we imagine then that they start growing. So now we have growth, but the growth is occurring in a group stage. So it's a new life history character. How growth in a growth in a group stage is a new life history character. But now when we get this group of four large cells, they can do two things. They can either separate, but if they're separate now, they're having to reorder these basic cell cycle stages, right? Normally, separation comes after division, but if we have growth and separation, that's a reordering of these life cycle stages. And so at first we thought, okay, well, this is a problem because reordering these stages is more difficult than just failing to separate and so on. So, but anyway, if that occurs, then we have this life cycle, and there's actually an algae that does that, Basiclamus. It's a four-celled organism, and it does that. And then, but the other thing is if we just stay with the order that was previous, what happens after growth in a normal climbing life cycle, you divide. So if that happens, well, before that happens, we have to recognize this problem of reordering. But forgetting, and there's a model that I won't go into that we've done to explain that. But the other is to just divide in the group stage and then separate. And this is the life cycle that all the algae do besides this one species that does this reordering life cycle. So we're trying to understand, why these life cycles have evolved, these two different kinds of group life cycles. But the origin of them comes from this failure to separate, we think. And so here's this beautiful organism, gonium. So it's reproducing, it's doing group reproduction. How does it do it? It's got 8 cells here. Each cell divides. So it's doing this life cycle here. And each cell divides, it becomes a big group, and then the groups separate. So it's doing this. So that's what tetrabena and gonium, that's the common group life cycle in this group. Most of our work now is on this organism because it's not committed to group living. There's no group boundary. They're held together just by little bridges, and there's no like group boundary. So under stressful conditions, they actually leave the group. So we're using that to try to understand more about this transient stage to committed group living, which does not exist in this organism. Good. Okay. Not going to go into this model, which explains the reordering of this life cycle, but not going to go into that for reasons of time. But I do want to make some general things. So our lab and one of my students was Eric Hanschen was involved in the sequencing of the genome of gonium under the supervision of Brad Olson, and what do we find? That the gonium genome, like I said, is just exactly like the unicellular ancestor, except this big diversification of all these cell cycle genes. And there's just these immense number of new cell cycle genes that had to evolve to get to this group life cycle. And so that's the gene family that really gave rise, we think, to this lineage of multicellular complexity in the algae. We're not going to spend a lot of time. We won't go through this, but we will say if we take, so there's this retinoblastoma pathway. So those of you that know a little bit about cell cycles, probably a lot more than me, I don't know, I know just a little bit. There's genes that that control passage through the cell cycle. And the RB gene is one of the key regulators that allows the cells to, after what is it, the synthesis, to proceed into the division phase of the cell cycle. So this is a master regulator, and it's diversified, part of this cell cycle diversification. If we take that gene from gonium and put it back into Chlamydomonas, we get groups. So that was quite a remarkable finding. So it causes cell groups. The evolved gene, we put it back in Chlamydomonas, and Eric was able to show that it creates groups in Chlamydomonas. And these are not the normal kind of groups that Chlamyd produces, which those of you that might know a little bit about this lineage, there's something called palminoids. Chlamyd is the cells stick together when there's stressful conditions. No, these are organized groups that are different from those palmoids. But anyway, wrap up of this stuff. And so what do we have? Wrap up in the group life cycle. Unicellular cycle has a transient group stage that can be co-opted and extended. This leads to the possibility of group-specific adaptations, and these group-specific adaptations then stabilize the group stage and allows for more complex adaptations to evolve. So let's move on to the final topic of individuality. But just then recognizing these three gene families that have evolved in this group, the somatic, Reg A-like genes, the cyclins to create the group life cycle. Then there's also something that I didn't talk about, the paraphorins that allow this space-filling stuff in the middle. And so those are the three differences, the three groups of genes that evolved in this lineage to create multicellular individuality. So in this slide, what do I have? I have the familiar criteria for individuality on this y-axis: genetic homogeneity, uniqueness, spatial boundaries, physiological unity, indivisibility, group level adaptations, division of labor, and the different MLS criteria, MLS1, MLS2. And then I have just on the top, just in increasing body size, the different kinds of algae that we study. And then you can fill in this whole table and ask, during this evolution in this lineage, which of these criteria of individuality has changed the most, okay? And so what we see are things like spatial boundaries, genetic uniqueness haven't changed much, genetic homogeneity not changing much. All the action is down here with group level adaptations, which allow the division of labor and these different the two MLS criteria to change. So, so that's interesting. So we can ask which of these in this lineage change the most, which of these don't. And to summarize that, we find that, individuality appears in grades. It's not a binary trait. Some criteria don't change much in the volvocines. I've already listed them. And some criteria change a lot. Okay, so now let me just jump into just how we use the word individuality in this theory. And I've already said it evolves in steps, it's not a binary thing. It's also not necessary for selection, right? I mean, this is what MLS theory shows us, that heritable variation in fitness can coexist at multiple levels, and that modifiers evolve to reduce the individuality at the lower level, or can evolve to reduce the individuality of the particles and increase the individuality of collectives. But it's not like a prior criterion for Lewontin's conditions. I definitely agree with that, and I think the multi-level selection theory shows us that. What are individuals in my usage? They're really the focal level of complex adaptations. and that the term is kind of dynamically earned its explanatory normative status out of the dynamics of natural selection. That is, we use it when we're at a level where below particle fitness has been regulated and most of the variation is at the higher level, and so complex adaptations can evolve at that level. And so, but it earns that status. It's not something that's imposed. The dynamics create that status. An ETI theory then is about the reorganization of individuality. It's not a theory about the origin of it in any absolute sense, right? You're not taking low individuality critters or entities and making high individuality um, out of the collectives of low individuality. I don't think that's possible. What ETI theory does is it takes collectives of individuals, creates new kinds of individuals. If you want to ask about the origin of individuality in an absolute sense, I think you have to go back to the origin of life, right? It's an origin of life question, how it first evolved. But the theory that we've been working on is about kind of the reorganization of individuality at different levels, which mobilize basically the individuality of the lower level. Another way of saying that is that the collectives become Darwinized by virtue of the Darwinian properties of their members. I'm not familiar with any kind of situation where that's not occurring. So we've been trying to translate this into the classroom, and I'll just say very little about that. I'm already over the time, but so we've been publishing in this space of literature where this is a model of how to do that, how to translate research into the classrooms. You have to identify the new knowledge. You have to translate it into the guidelines that are locally operative in the school. And then you have to develop tools. So we've been doing that. We've been publishing in this space about these different phases, and I'll just advertise that. And won't say any more about that. But if you're interested in K through 12, what's called K through 12 in the US, education, I think ETIs are a good way to teach complexity because it has this social fabric. And why do that? Well, because it teaches the hierarchical organization of the world. See, that's something that's not ever taught. It's kind of assumed, okay, there is this hierarchical structure, but where did it come from? And so you have to, I think teachers, that should be taught. You should teach that the world is complex in this hierarchically structured way because of these major evolutionary transitions in individuality. And also so it deepens the knowledge of biology, but then it also connects the science to the lives of students and being social critters. Good. Okay, I think I'm going to wrap it up there. Thank you for your attention over time here. Good. So that's hope you're still there and I hope the technology is still working.
Absolutely. Thank you very much, Rick. That was great, and we did follow everything. So that's absolutely perfect. So we have time for discussion, and Rick was clear that he wanted questions, comments, and critiques. So let's see how. So I see one comment in the chat. Please, I mean, the easiest thing would be, if that's okay for you, if you just raise your hand virtually so we can see you. Let me see, maybe. Okay, good. I can see everyone now. So. Duncan, would you agree to ask your question orally? Maybe that's going to be easier for everyone. Oh yeah, thank you.
Hi, can you hear me?
Yes.
Hi, Rick. Thanks for the great talk. Super fascinating learning about it. So the question, do you think the limited evolutionary expansion seen after the volvocine transition reflects the fact that it involves coordination within a single clonal lineage, whereas transitions based on cooperation among non-related individuals, they integrate genuinely heterogeneous capabilities and therefore open much larger evolutionary search space, i.e., why does clonal aggregation consolidate while endosymbiosis explodes?
Yeah. Yeah.
Hang on, I'll mute myself. I can't hear you.
Yeah. Can you hear me now? Or yeah. So you're bringing up this question about, fraternal transitions and um, egalitarian transitions. And so, they both have different challenges. So like you say, the challenge for a fraternal transition where you have a lot of kin selections, how do you get diversity within the group? Um, um, Whereas the challenge, but however, the alignment of fitness happens naturally out of the kinship. Whereas in a egalitarian transition, I don't know if you're familiar with these words, but within like endosymbiosis, you have um, interesting, diverse functions brought together, but the problem is to align them, and so that the group can proceed. So I think, these two kinds of transitions have different challenges. So back to the Volvox, you ask, okay, is the limitation on um, the diversification of the lineage because they're starting with kin groups? And I think that's probably not, I mean, a counterpoint in disagreement to that is that, our bodies, we're the most complex group, but we originate from a single mitotically dividing zygote, right? And there's a lot of diversity created in that, in our bodies, maybe 200 cell types. So I think that's probably not the answer alone. Yeah, but I'm open to your thoughts.
Thanks so much, Rick. It's a lot a really interesting subject to think about. So I'm going to continue thinking about it, but really appreciate your answer. Thanks a lot.
Rick, I have a quick follow-up on this. What do we know about foreign material in volvocine in the sense that what do we know about pathogens? What do we know about the microbiome-like things in volvocine? What do we know about resident viruses? For example, in the work of Toby Kears, also on transitions, the foreign material is very important, and this is very much related to your question, Duncan. So, what do we know about that? Is there some foreign material in volvocine or not?
We don't know much about that. Yeah, it's a good point, and so I'm ignorant on the reference you gave about work that identifies this as being important. I'm not familiar with the work you cited. Um, if you could expand on that, but I would like to know about that, but I don't know this reference you gave. Maybe you could put it in the chat, then I could click on it and learn about it.
Yeah, yeah, and we can talk later about that. It was just a quick follow-up, and this is not my role to anyway to ask questions. I see that Pierre-Henri, good to see you, Pierre-Henri. Pierre-Henri has a question.
Hello, Rick. Thank you very much
Hello.
for this talk. Long time no see. Yeah.
Right.
I wonder whether you had tried to integrate the selfishness in your model with like cancerous cells, for instance. Have you tried to integrate that, and do you observe things like that in Volvox, for instance?
Um, I didn't hear you clearly, Pierre-Henri.
Ah,
The question was, I just want to make sure I understand your question. Do we have things like cancer and Volvox? Was that the question?
that's the end of the question. But the first one is, in your model, do you integrate selfishness like cancer cells or things like that?
Well, just in the sense of this Reg A gene, when there's a mutation in it, the cells just stop, they start dividing. So they disobey the group level coordination where that requires them to not divide. Is that
Yeah,
your,
yeah. Well,
and that creates this cancer-like phenomena in the where there's just but it's not like a cancer tumor in the colony. It's like the whole colony becomes a tumor and just doesn't do any cooperation.
but you've not found localized aspects like that. That is not
spatially localized, you mean?
Yeah,
Spatially, yeah, no, we haven't seen that. Yeah.
Anton.
Thank you. I've got a theoretical question. Unfortunately, I was not familiar with your work before. It was very interesting because I did something similar. So I just glanced on it today in the morning. As far as I understand, behind it, the assumptions are the same that we have in the price equation. Additivity of fitness or non-linearity in general, and no epistasis, no things like that, complications. And I understand this choice, but is there any indication, maybe simulations, whatever, showing that the conclusions survive when these complications are added back? Thank you.
Yeah, so yeah, I think no. So you're worried about just the simplifying assumptions that the conclusions are a result of the simplification, right? Or in general, which assumptions are you most worried about?
Lack of epistasis, non-linearity in general.
Yeah, the equations are so nonlinear, it's hard to imagine wanting to add more to them. So there is a gene it's a two-locus model, and the effect of the modifier does depend upon whether it's in a cooperative background or a defection background. So there is epistasis, and it's just built into the model. So there's this like cooperate defect game being played at one locus, and then there's the modifier that changes the structure of the mitotic lineage. How for example, how in a germline you might sequester some cells, they'll divide for a few replications, but not as much as the soma. So there is epistasis, but it may not be the kind of epistasis you're looking for. I don't know. Yeah, I mean, you wouldn't. So no, I haven't done simulations of more complicated stuff. I like the pencil and paper kind of mathematics. That's where I work. But I think should be computer models, more complex things. I suppose there are things in the literature that I don't know about.
Do we have other questions? Also, maybe coming from philosophers. As you saw, Rick was both very biological and very philosophical today, especially in the final part. So I expected some questions about individuality. I have a very general question about this, which is something I've seen again and again, but always strikes me as very implausible. Some people say that we should try to get rid of the notion of individuality altogether. And I was thinking that because of what you said about Dick Lewontin talking about fitness being so special to biology, maybe that's the reason why we cannot get rid of this notion of individuality in biology. I just wanted to have your opinion, Rick, on that. Could we do without it?
No, I don't. I think we have to count things first of all, right? If we're going to operationalize this theory, we have to go out and count stuff. I mean, that's the first point there. I think you have to count things if you want to like measure natural selection. So what is it you count? They're individuals. So that's the first point there. I think the second point is that what do I want to say? I'm not a philosopher, but I've been interacting with my AI friend, and the AI bot says I'm committed to when I feed it my work, the AI bot says that I'm committed to a normative explanatory notion of individuality because it's something that's earned by the dynamics. And it's something that only can give rise to complex adaptations. I'm curious on feedback on that point from philosophers. I was struck by the word normative because I was, I don't see this as, value laden. It's just the dynamics of natural selection. But I guess the word normative can be used to recognize certain kinds of explanation that are privileged. And so I think the individual level is privileged in the sense that and only it can evolve this complexity that I'm interested in. But I'm happy to have feedback on that.
Okay, I see that there's another question by Anton, but first maybe I see Marie has a question, so let's try to have, and it seems that she cannot turn on her microphone, if I understand correctly. So she asked on the chat, do you think that the evolution of animal coloniality with more or less integrated, specialized individual animal could rely on similar mechanisms?
I do, yeah. So I think the generality has to do with this life history framework that that in the choanoflagellates, the unicellular stage will have similar genes, life history genes that turn off reproduction in stressful environments. I can't imagine any kind of living thing not having that. And so if they have those genes, then they can be, well, in principle, they should be easily co-opted to be turned on in a spatial context in a group and to create soma. So I do think that, but I don't have any evidence of that. But I would predict that. Yeah. Thank you for that question.
Okay, thank you. Anton, did you want to ask another question? Okay, maybe Anton. Okay, Fridolin has a question.
Yes, hello. I have a question. It's more of a curiosity about something you said in the beginning when you described adaptation. You said you think of it as an outcome of a kind of self-organization process in a population dynamical system, and that sounds a bit like a non-standard formulation of the process. So I was wondering if you could elaborate on that. Thank you.
Yeah. Well, what I was trying to get at there, but Dan Dennett has this way of talking about, that this kind of backwards explanation. I forget how he phrases it that exists in biology that to make a complex and beautiful machine, you do not need a more complex and beautiful machine. So everything before Darwin was, to make something beautiful like a watch, you had to have something more complex like a human. Whereas Darwin reversed that logic. So okay, so if we accept that Darwin reversed that logic, that adaptation can emerge out of population dynamics. That's all I think I was trying to say. Maybe the word self-organization was a misuse of that term, but it's just if there is if it's just natural selection is all I was trying to say, right? If there is there's variation in the population and that if there's some of those traits are heritable and some of those traits benefit the reproduction or survival of the entity, then those traits will increase. So it's just that process of natural selection. I was just trying to encapsulate that when I said self-organization, but maybe that was confusing. Yeah.
Okay, no. The reference to Dennett made it clearer. Thank you.
I forget what he said. He said it more eloquently than I said it, but I can't quite remember. Yeah. There was some critique, I guess, at the time of Darwin that criticized Darwin for having this reversal of logic, right? And I forget the guy's name. Dennett makes a big deal of the critic of Darwin that said that. that Darwin's asking us to believe that to make a beautiful and complex machine, we don't need something more complex. But he hit the nail on the head, but he thought it was implausible, but does that help?
Fridolin, do you want to follow up, or you said
No,
everything?
thank you. Thank you. No,
Okay,
that's
okay. I just wanted to be sure. Okay.
Sorry, I missed the last part. Thank you.
I don't know if I'm pronouncing this right. Jaren? Or the person who raised your hand, could you please ask your question? Oh,
Yes, if I
ah,
can ask a question. Thank you, Rick, for this nice talk. So perhaps elaborating a bit on what Thomas was saying before, he was mentioning parasites and that kind of organisms which are probably interacting with the volvocales. But I was also thinking more generally about the general environment. They are probably predators. They are living in moving environments. And do you think that they are, some kind of interactions with other living beings or with the environments which are favoring the evolution of multicellularity in general? Because I mean, you focused on volvocal, but there are many examples of evolution of
Yeah.
multicellularity.
Well, in this lineage, it's thought that predation was important. Bigger things eat smaller things, so there's an advantage to being bigger with regard to avoiding predation. So in the lab, with these lineage, like with Chlamydomonas, people have evolved multicellular forms by putting them in environments where there's predators. And so that I think is it's the most widely discussed in this lineage, external kind of selective force for multicellularity is predation. Yeah.
Rick, could you please elaborate very quickly on the ECM? You said that it was a great way to cooperate, and I agree, but you also seem to say this is the best way to cooperate, or maybe the only way to really cooperate. And I was not so sure about this. Could you maybe elaborate a little bit on the centrality of the ECM in your thinking?
Yeah, I didn't mean to say it's the only way to cooperate. The most common way of cooperating is the flagellar motion. There are these interactions between cells in the boundary that allow the bigger colonies to move faster than the smaller colonies. So I think motility is probably the most is a important form of cooperation. And interestingly, in the algae, there's not a central coordination of the motility. So you have cells spread around a sphere, and their flagella have to be at different angles to get optimal movement. And the coordination of those angles has to do with the local dynamics. I mean, this is the work of hydrodynamic physicists have shown this, that there's local dynamics about how the flagella interact on the sphere that creates the coordination of the flagella. Like there's not a brain that in the algae. So anyway, I think the cooperation among the flagella is important. The ECM, it just seems that it ought to be a kind of a commons, right? A kind of place to store nutrients, but little work's been done on that. It's more of an interpretation that we place on the diversification of the paraphorans that create the ECM. Rather, the interpretation is that it's for cooperation. That hasn't really been tested. Yeah. So I probably oversold that comment. Yeah.
No, it was very interesting, and the clarification is very useful. And maybe I overinterpreted what you said, so no problem. It's very useful clarification. I see we're almost at the end of the time we had. Is there any pressing question or comment for Rick before we thank him again and say goodbye? I don't see any questions in the chat. No hands raised. So, okay. So, really, Rick, it was absolutely fantastic, illuminating. Thank you so much. And I hope to see you soon. And again, thank you so much for the talk and the discussion.
Could I have a follow-up conversation just with you, Tomas, just about
Sure,
some
sure.
practical
So we're
stuff?
staying and the others can leave if this is okay for everyone. Thank you very much,
Well,
Rick.
Goodbye.
thank
Thank you.
you. Pierre Henri, take care, buddy.
Yeah,
Good to see you, man.
yeah, right.